Ladies and gentlemen, you are all welcome to my YouTube channel. Today, we are going to look at the concept of cell structure and function for distance learning student, and this is our second lecture series. And of course, my name is Mokhtar Adiyu from the Department of Biochemistry, Ahmadu Bello University, Zaria. So let's start with the introduction. What is a cell? A cell is the structural and the functional unit of life. Of course, cell is what determined the life and deeds of each and every living organisms. If your cells are alive, it means that you are alive. If your cells are dead, it means that you are dead. And of course, this different cell, these cells is what determined the disease and the health status of each and every living organisms. So cell is what determined the life, the health, the disease condition of each and every living organisms. So, and of course, all living organisms or all life material on earth is due to the position of cell. So if there is no cell, there is no life. So cell is what determined the life of each and every living organisms. And of course, each and every living organisms on earth is made up of cell. So cell is made up of every living organisms. So if you see any living organisms or anything that actually have or that possess life, just because of the cells. So billions and trillions of cells combined and joined together to produce a complete living organisms. And of course, in humans, we have over 220 different cell types. Like for example, in average human being, there are over 37.2 trillion cells. So in average human being, there are a total of 220 different cell types. And each of these cells have their specific functions and they have their specific structure. So there are 220 different cell types. And these differences in each of every cells in human being are due to differential gene expression. What is differential gene expression? Differential gene expression is a process that involves the translation of gene into protein. So when you have gene, which is from the DNA, a gene is a segment of DNA that have the ability or potential to produce a specific type of protein. And of course, each and every cells have different structure and different function due to production or expression of different proteins on those different cells. Like as I said, we have over 220 different cell types. So what makes these cells differ from one another is because of the proteins that are produced from those cells. Like for example, the red blood cells is biconcave in shape and it is responsible for the transport of oxygen from one part of the body to other tissues of the body. And it's what makes red blood cells to have that biconcave shape. The transportation activities of oxygen to various organs of the body is due to the presence of hemoglobin. And of course, in humans, we have a specific genes that is responsible for producing that hemoglobin. And that's what makes red blood cells to be red blood cells. And of course, in the pancreas, we have proteins that make pancreas differ from the red blood cells. Like for example, in the pancreas, we have insulin, glucagon, somatotrophin. These are the types of the proteins that are produced in the pancreas, and that is why pancreas is differ from red blood cells in terms of function and structure. So different proteins that are expressed or that are produced from different genes on different cells is what make one cells differ from one another. So that is why, like in human beings, we have like liver cells, which is hepatocytes. We have nephrocytes, that is the cells of the kidney. We have neurons, that is the cells of the brain. And of course, we have sperm. We have sperm. So these are the different cell types that are found in humans. So it means that in human being, there are 220 different cell types. Examples like the liver, like the pancreas, and many other different cell types. So we have over 220 different cell types, and these differences of cells is due to differential gene expression, and this differential gene expression is what actually leads to expression of different proteins. So, and then in addition, we are going to look at now scientific discovery of cell. So the first scientist that discovered the cells is Robert Hooke in the year 1665. He accidentally discovered cell. So this Robert Hooke was actually the first scientist that was able to discover cell accidentally in the year 1665. Then followed by Anthony van Leeuwenhoek. He is the first scientist that was able to observe spermatozoa, human cells, and bacteria in the year 1675. So it means that after the discovery of cells by Robert Hooke, he was able to even publish a book called Micrographia. And of course, this Robert Hooke was the first scientist that was able to name the cell. So he's the one that named the cell as a cell. And of course, after that, then is the Anton van Leeuwenhoek. He is actually the first scientist that was able to clearly demonstrate or that observe spermatozoa, human cells, specifically erythrocytes, and the bacteria in the year 1675. In fact, in microbiology, Anthony van Leeuwenhoek was considered as he was actually one of the great pioneers of the area of microbiology. So he made a great contribution by being the first scientist to observe bacteria using a compound microscope, which is a little bit more sophisticated and powerful than the microscope discovered or used by Robert Hooke. So then the next actually set of scientists is actually Mathis Kleiden and Theodor Schwann. They are the scientists that was able to come up with the cellular theory. And of course, this Mathis Kleiden in the year 1839 and the Theodor Schwann, they are the one that was able to come up with the cellular theory. So all organisms, the first cellular theory stated that all organisms are made up of cells. So it means that all living organisms, every living cells or every living organisms are made up of cells. And of course, these cells are fundamental unit of life. And of course, the function, the health and the disease status of each and every live organisms depend on cells. And if there is an injury in a cell, it means that there will be a disease. And of course, it depends on the types of the injury. It can be reversible injury, it can be irreversible injury. So in the case of reversible injury, it's a type of injury that's the disease can be cured, or the cells can actually still maintain or can still regain its normal physiological activity. But in the case of irreversible cell injury, it means that the cells can actually proceed to death. The cell can die. So if the cells are dead, it means that the tissue will also be dead. And if the tissues are dead, the organs will be dead. And of course, the organs that are actually dead will also affect the system that is actually work with that organ. and which may actually subsequently lead to death. And of course, that is why we have cellular replacement, that is cellular division. So cells divide as some of these cells are dead, then new ones will be generated. So, and of course, the death of the cells can be necrosis or apoptosis. So if it is apoptosis, it means it is a programmed cell death. And if it is necrosis, it means that it is unwanted cellular death. And that can actually lead to disease in many cases. And then in addition, which is actually the third cellular theory, is that all cells come from free existing cells. Like, for example, we as human, we come off actually from human cells. Dog cells are coming from actually dog cells. Although there are evolution which can actually lead to speciation, but all cells come from free existing cells. You cannot get cells from stone. So therefore, actually, these scientists have made a great contribution in actually the discovery of cells using microscope. But of course, with a lot of technological advancement in science, more discovery have been made in the area of cellular structure, which we actually going to look at them as we continue with the lecture. Ladies and gentlemen, I always advise you, if possible, try to join the YouTube channel, and at the same time, you can also visit our website, rafisch.com.ng. Because if you actually joined our YouTube channel, of course, we are going to have a lot of other videos which can only be viewed by only members of the YouTube channel. So then we are now going to look at the classification of cells. Cells are classified into unicellular organisms. So when we said unicellular organisms, it means that there are organisms that have only one cell. And examples of these organisms that have one cells, we have bacterial cells. We have like Framicium, Amoeba, Euglena are the examples of organisms that have only one cell. So this is actually a typical example of bacterial cells. This is a structure of bacterial cells, which is one of the examples of unicellular organisms. So apart from the unicellular organisms, the next classification of cell is multicellular organisms. So generally, multicellular organisms are organisms that have many cells. They have many cells. Like examples of organisms that have many cells, we have plant cells, animal cells, we have fungi. So all these are the examples of multicellular organisms. And of course, examples of multicellular organisms, we have human. So generally, humans have billions and trillions of number of cells. Like, for example, it has been estimated in average human being, there are over 37.2 trillion cells. So average human being contain an approximate of 37.2 trillion cells. So this shows that humans is a typical examples of multicellular organisms. And each and every cell has its own lifespan, especially in humans. With over 220 different cell types, each and every cells in the body have the ability to actually, although it's not all, there are some cells that after some time, when the use of the needs to be dead and then replaced with another one. Like, for example, let's look at these different cell types and their lifespan, as well as the cellular division. Like, for example, this is esophagus lining. Esophagus lining is a cell that actually takes 2 to 3 days to be replaced. So after 2 to 3 days, these cells will die. And then, of course, they will be replaced. That is why here you can see clearly they can undergo cell division. It means that after these 2 to 3 days, the esophagus lining will die. And then, of course, the new ones will be generated through cellular division and then replace the dead one. Then, of course, we have small intestinal lining. There are also cells of the small intestine, and after between 1 to 2 days, they die and then they will be replaced with the new ones because the cells can undergo cellular division. So they will be divided and then the new ones will be generated. And there is also a large intestinal lining which takes them 6 days, and after 6 days, these cells will actually be divided and they will be generated and then replace the large intestinal lining. And then at the same time, the red blood cells after 120 days, they will die and then they will be replaced after that 120 days. Of course, the red blood cells, they are produced in the bone marrow, and then these red blood cells will now replaced with the new ones. So then we have white blood cells. So white blood cells, they take like 10 days to decade before we are able to actually die. So after the date, then they would also be replaced, because although many don't divide. So of course, the muscle cells, the skeletal muscles and the nerve cells are the typical examples of the cells that most of them, they are the cells that we bond with and we dead with, because their lifespan is long. They have long lifespan. In fact, in many cases, if you can check some other textbooks, they will tell you that these cells do not die until when you are going to die completely, because as these cells die, because they are not undergoing any cellular division. So after they die, you cannot replace them. You cannot replace them. So if you lose your heart muscles, if the only way is actually to do heart transplanting, to transplant new heart, because the cells there cannot divide. The skeletal muscles also, the nerve cells, that is the brain cells, they also take longer time. In fact, as I said, in so many other textbooks, they will tell you that they don't have lifespan. It's just that once these cells are dead, then you are also dead, until or unless if you are going to undergo tissue transplanting or organ transplanting, like for example, brain transplant, skeletal muscle transplant, or heart transplant, because none of these cells undergo cellular division. So as they dead or as they have any injury, there will be a problem. So this is actually a different lifespan of human cells. So human cells have different lifespan, although it's not only in humans, other organisms have their different cells as well as their lifespan. So then the next thing is actually we should understand that in animal cells have different shapes. They have different size. Some are circular, some are spherical, some are cylindrical, while some are fibrous. So we should understand that animal cells, they have different shape, they have different structure, they have different sizes. So cells do not have the same size. So different cells have different shape. Like sperm cell is the smallest cells in animals, while ova, which is the egg cells, is the largest in humans. Then in terms of length, then the nerve cells are the longest. So we should understand that the smallest cells in animal cells is sperm cell, and then the largest cells is ova, and then the longest one is the nerve cells. So we have different types of cell. We have prokaryotic cell and we have eukaryotic cells. So what is prokaryotic cells? So generally prokaryotic cells, as we can see clearly here, they don't have nucleus. They don't have membrane-bound organelles, and they are smaller in size. They are very simple, and they are unicellular organisms. So most of the prokaryotic organisms, they are unicellular, they are very simple, they are smaller in size, then they do not have specific organelles like mitochondria, Golgi body, and the rest, and there is no specific nucleus. And then for the eukaryotic cell, the nucleus is present, and the eukaryotes, they has organelles, they are bigger in size, they are more complicated, and then they are multicellular organisms. So these are the differences between prokaryotic and eukaryotic, and at the same time there are similarity. So in terms of similarities, they all have cell membrane, they have cytoplasm, and they all have DNA, and then they all have ribosomes. So these are the some of the similarities between prokaryotic cells and eukaryotic cells. So then, of course, the next thing is cellular component. So cells have different components. So there are different components of cells. We have cilia, we have lysosomes, mitochondria, rough endoplasmic reticulum, cytoplasm, nucleus, Golgi bodies, microtubules, and smooth endoplasmic reticulum. And with time, we are going to look at the different functions of each of these organelles. But now we are going to start with the cell membrane. So now, what are the functions of cell membrane? The cell membrane are also called plasma membrane. So plasma membrane is another name for cell membrane and is found in all cells. It is found in all cells and it separates the interior of the cells from the outer membrane. So generally cell membrane, it separates the interior of the cells from the exterior. Like for example, this is the outside cell membrane and this is inside cell. This is outside cell and this is inside cell. So cell membrane is what actually separates the inner part of the cells from outside of the cell, from outside environment. And then the cell membrane, it consists of a lipid bilayer. So the cell membrane have a lipid bilayer. This is the first layer and this is the second layer. So that is why it is called lipid bilayer. This is the lipid bilayer that is semi-permeable. It means that when we say semi-permeable, it means that it's not everything that can get into the cell. So it controls what will get into the cell and what will actually not get into the cell. So it controls the transport of substances that will get in and out of the cell. Then the next thing actually is the cell membrane regulates the transport of materials entering and exiting the cells. That is what will get in and what will get out of the cell. And of course, we have different types of membrane transport. So a cell membrane have different types of the transports. We have simple diffusion, facilitated diffusion, we have active transports, and then we have so many of them. So simple diffusion is a type of diffusion in which lipophilic substances or molecules can just get into the cells directly without using any protein carrier or without using any transporter. So that is in the case of simple diffusion. And of course, a simple diffusion is the movement of molecules from the region of higher concentration to the region of lower concentration. You can see clearly here, this is the molecules. So the molecules, the concentration of molecules here is higher compared to here, because here, if you can see clearly, it is only one molecule. So the concentration of molecules is higher here. So the movement of molecules from the region of higher concentration to the region of lower concentration is what we call simple diffusion. And then the next one is facilitated diffusion. A facilitated diffusion is a types of transport in which molecules are transported into and outside of the cells with the help of protein carriers and protein transporter. So it needs protein transporters. Then for the active transport is actually a transport of substances from the region of lower concentration to the region of higher concentration using ATP. It uses ATP to transport substances from the region of lower to the region of higher concentration, while for the facilitated diffusion is also from the region of higher concentration to the region of lower. Also in the case of simple diffusion, it is actually a movement of substances from the region of higher concentration to the region of lower. So simple diffusion and facilitated diffusion, they doesn't require energy, and of course simple diffusion do not require any protein transporter, while facilitated diffusion need protein transporter. So this is actually how some of the particles get in and out of the cells. So the next thing is actually the plasma membrane or the cell membrane. It provides a protection for a cell. So because it is like a cover that covers the cells and it protects it from any external attack. And then the next thing is also very important in the transport of nutrients into the cell and also transport of toxic substances out of the cell. So nutrient, when we take nutrient like glucose, amino acid, they will get into the cell using a cell membrane, and then toxic substances that can cause danger in our body also exit the cell. They will be transported out of the cell. And then the next thing now is we are going to look at the function of cell membrane. So cell membrane have many functions, including one, protection. So the cell membrane, it protects the cell from its environment by acting as a barrier to keep the cell's content in and unwanted substances out. So cell membrane gives protection to the cell by controlling what will get in and what will get out of the cell. So this actually give protection to the cell. The next thing is the transport. So cell membrane regulate the transport movement of substances in and out of the cell, and they allow essential nutrients to enter the cell and waste product to exit. And then second, communication. So one cell can easily communicate with another cell, and also can communicate with other substances out of the environment. So the communication of the cell is through the cell membrane. So cell membrane allows cells to communicate with each other and then respond to changes in their environment. Like for example, if a cell is actually infected with one microorganisms, then the infected cell will now communicate with the nearby cells within the cell membrane to actually alert that cells that there is a danger, so that it will now purify itself against the danger that affect that particular cell. And then structure, the cell membrane also provides structural support for the cell. And then compartmentalization, in eukaryotic cell membrane define the internal compartments such as the nucleus and the organelle. So it provides the compartmentalization, nucleus, mitochondria, Golgi body, smooth endoplasmic reticulum, they are all compartmentalized. So cell membrane actually provides that compartmentalization. Like for example, the nucleus is a membrane-bound organelle, so it has its own membrane. So it actually compartmentalizes with other organelles. So cell membrane of the nucleus can give compartmentalization or can provide compartmentalization with other organelles. And then, of course, the next thing is, let's look at the cell membrane structure. So in the cell membrane structure, we have the prime part. We have carbohydrate sugar chain. We have hydrophilic heads. We have cholesterol. We have phospholipid bilayer. We have hydrophobic tail. We have integral proteins, and we have peripheral proteins. And then we have cell membrane receptor proteins. So now, we are now going to look at, and of course we have glycoproteins, and then we have globular proteins. So we are now going to look at the functions of each of these organelles of also the different component of the cell membrane, because they are very essential. Like for example, when you said hydrophilic heads of the membrane, it means it's a region that can interact with water. Philic means love, so it can interact with fluids. And then hydrophobic, from the word phobia, the hate fluids, the hate to interact with water. So we are now going to look at their functions one after the other, and then their relevance in the cellular structure and function. So the first thing that we are going to look at is phospholipids. So phospholipids are one of the principal components of the cell membrane. So it's one of the major components of the cell membrane in conjunction with membrane proteins. So generally, these phospholipids, they are they are the most abundant component of the cell membrane, and they usually also combined or associated with the membrane proteins. And these phospholipids, they are amphipathic molecules. When we said amphipathic molecules, it means that they can actually have both hydrophobic and hydrophilic compartment. When we said hydrophilic, it means that it has a component that can interact with water, and also hydrophobic regions, that is a component that cannot interact with water. So amphipathic, it means it's a molecule that have both the water-loving region and the water-hating region, that is hydrophilic and hydrophobic, or polar and non-polar. So the next thing after this is Let's look at typically the structure of this phospholipid. So this is actually a structure of phospholipid. It has a polar head, and this is a polar head. That is the organic molecules that can usually attach to these polar heads. They are organic molecules which can be serine, which can be choline, which can be phosphates. So therefore, these polar heads can interact with water molecules. And then there is a non-polar head. The non-polar head or non-polar tail is called hydrophobic. It means that it cannot interact with water. And this non-polar tail or hydrophobic compartment is made up of two important things. It is called diglyceride. Why is it that it is diglycerides? It's because there is one glycerol and two fatty acid. So in the non-polar tail of the phospholipid, it has one molecule of glycerol and two molecules of fatty acids. And then in the polar head, we have organic molecules which can be serine, which can be choline. Like for example, a polar organic molecules, e.g. can be choline, can be serine, and then of course a phosphate. So this is the first component, a polar organic molecules, and then the second component is phosphate group, and then the last component is glycerol molecules, which is replaced by sphingosine in the case of sphingomyelin. So these are actually the component of the phospholipids of the cell membrane. So then the next thing is cholesterol. So what is cholesterol? Cholesterol is another lipid component of animal cell membranes. The cholesterol are selectively dispersed between membrane phospholipids. So it is selectively dispersed. So you can usually find this cholesterol around the membrane of between the membrane phospholipids. So it's usually found in between phospholipid and the cell membrane. And then this cholesterol, it helps to keep cell membrane from becoming stiff by preventing phospholipid from being too closely packed together. So it helps the cell membrane from becoming stiff. And this is due to preventing the molecules of the phospholipids to closely packed together. And then in addition, the cholesterol is not found in the membrane of plant cells, which is very interesting. So what are the functions of this cholesterol in the cell membrane? Number one, it is actually a precursor of vitamin D, and it's also important in plasma lipoprotein constituent, and it is also a precursor of steroid hormones. So with cholesterol, you can actually synthesize steroid hormones like aldosterone, cortisol, progesterone, estrogen, and testosterone. So these are the kinds of the hormones that can be actually generated from cholesterol. It is also served as a it's also used in bile acid biosynthesis. It also important in insulating nerve fiber. And the most important functions of the cholesterol relevant to our topics today, it's actually important in membrane fluidity. So the membrane is actually fluid in nature according to fluid mosaic model. So the cholesterol is important in maintaining the membrane fluidity, and it also have in membrane homeostasis. So these are the two important functions of cholesterol in relation to our today's topic. So then the next thing after cholesterol, of course, is glycolipids. So glycolipids are located on the cell membrane surface and have a carbohydrate sugar chain attached to them. So glycolipids are those lipids on the membrane surface, and they have a carbohydrate sugar chain that attached to them. They help the cell to recognize other cells of the body. So you see, this is actually the membrane. So if you look at the cell membrane, in the membrane there are lipid residue, and this lipid residue joined with a sugar residue on the surface to generate or to produce the glycolipids. So this is how the glycolipids are produced, and it's very, very important in cellular recognition. It's very, very important in cellular recognition. That is, to recognize nearby cells, to recognize unwanted cells, to recognize microorganisms. So glycolipid is very, very important in doing that. And then the next thing is actually the membrane proteins. So we have two types of associated membrane proteins. So we have two types of membrane proteins. We have peripheral proteins and we have integral proteins. So in the case of peripheral proteins are exterior to and connected to the cell membrane by interaction with other proteins. So in the case of peripheral proteins, you will see that they are just transiently or partially attached to the cell membrane. Look at them here, they are partially attached to the cell membrane. And examples of these peripheral proteins, they are enzymes that are found on the cell membrane. They are also anchorage proteins as well as the transporters. So most of the transporter proteins and the carrier proteins, enzymes and anchorage proteins, they are peripheral proteins. And then for the integral proteins, they are inserted in the cell membrane and must pass through the cell membrane. So it is actually fully penetrate. It fully penetrates into the cell membrane. It is fully penetrate into the cell membrane. So these are the examples of these integral proteins. And in addition, so these integral proteins, they are involved in recognition, cellular recognition, and some of them, they are receptors. There are also enzymes, like for example, the integral protein can be alpha helix, can be helical bundle, can be beta barrel. So these are some of the examples of the integral protein. It can be alpha helix, helical bundle, and beta barrel. So the beta barrel can be transporters or channel proteins, and then the helical proteins can be transporters, receptors, or enzymes. So the next thing after these cell membrane proteins is, of course, we have discussed about the different types of membrane transport partially on our previous slides. And of course, we are going to specifically have a lecture on the different types of transport of substances across the cell. So thank you for listening. Thank you very much for listening, and thank you for joining thank you for subscribing and joining my YouTube channel. And if you did not join my YouTube channel, please join, because we are going to have so many added advantage. You can have, of course, there are so many advantage. Like for example, we are going to be setting some questions that are related to our lectures. So thank you very much. Thank you very much. And of course, if this is the first time you are coming, please subscribe and join. And you can also visit our website at rafisch.com.ng. So if you joined, we are going to have so many other lectures related to physiology, related to medical biochemistry, related to microbiology, which will be actually very important. And of course, it will be important to you. So thank you very much. Thank you very much. Bye bye.